What is polypeptide synthesis? Polypeptide synthesis is the process of biological production of peptides, which are organic compounds in which multiple amino acids are linked via peptide bonds. These peptides can consist of between 2 to 50 amino acids. Peptides are short chains of amino acids connected covalently by peptide bonds that act as building blocks of essential proteins such as collagen, elastin, and keratin. These proteins provide skin with optimal elasticity and firmness. Peptides are significant in the construction of many other proteins which are crucial to growth, repair, structure, function, and regulation of body tissues and organs, and therefore the survival of the organism. The overall process of polypeptide synthesis can be broken down into two key steps: transcription and translation. Protein synthesis occurs outside the nucleus. Therefore, it is crucial that an exact copy of the DNA is not sent beyond the safety of the nucleus walls into the cytoplasm. In order to prevent this, transcription takes place within the nucleus, where DNA is located, to create a template of the DNA needed for polypeptide synthesis. This new molecule is called mRNA, messenger RNA, and differs from DNA in a variety of ways. mRNA is single-stranded rather than double-stranded, meaning it doesn't form helices, but rather folds into complex structures that are stabilized by internal complementary base pairing. mRNA's sugar component is a ribose rather than a deoxyribose, and it also consists of uracil nucleotides instead of thymine nucleotides. The created mRNA molecule is labile and more susceptible to hydrolysis, which allows the molecule to dissipate quickly in the cytoplasm after the completion of protein synthesis. The transcription process takes place in 3 steps. Step 1, initiation, the decoding of a cell's genetic information. An enzyme helicase in an ATP-dependent reaction attaches to a section of the gene known as the promoter, located near the transcription start site of the gene. This binding of the helicase indicates and influences the unwinding of the DNA in order to allow the bases of one of the strands to be read. The enzyme RNA polymerase then binds to the unwound DNA and decodes the entire strand, now acquiring the ability to construct a strand of RNA with a complementary base sequence. Step 2, elongation, the addition of nucleotides to the RNA strand.
Following the enzyme's base reading, an mRNA molecule is built using the complementary base pairs. A 5' is added to the beginning of the RNA transcript, and a 3' poly-A tail is added to the end. This cap and tail protect the transcript and assist in the exportation of the strand from the nucleus and translation onto the ribosomes. During this process, there is a brief time when the newly constructed strand is bound to the unwound DNA. In this time, an adenine nucleotide in the DNA strand binds to a uracil nucleotide in the mRNA strand. This means the mRNA is not an exact copy of the DNA. Step 3, termination, the ending of transcription. The enzyme RNA polymerase continues to decode and construct until it crosses a termination sequence in the gene. This signals that the mRNA strand is complete and it detaches from the DNA strand. The mRNA strand created provides the specific codes and information of the exact order of amino acids required to bond together in order to form a polypeptide. RNA splicing, the removal of introns from the mRNA strand. In gene strands, there are portions of the gene that do not code for amino acids. These are known as introns and are removed from the mRNA strand in a process called RNA splicing. Exons are portions of the gene that do code for amino acids. Name this as they are expressed in the proteins. Introns are in between the exons and, in order to create a mature mRNA strand, each end of the exons must be brought together and the intron eliminated from the strand. RNA splicing is a process in which introns are recognized and removed by a protein and RNA complex called the spliceosome. All introns must be removed from the mRNA strand. If the spliceosome fails to remove an intron, an incorrect protein will be produced during translation. After the intron has been removed, the ends of the exons are brought together to form a mature mRNA strand that can now correctly code for proteins. Translation is the process of the synthesis of a protein. After transcription is complete and the mRNA strand has detached from the DNA strand, it is transported from the nucleus through the nuclear membrane and into the cytoplasm, attaching to a ribosome that is made up of rRNA. Within the cytoplasm, there are many tRNA molecules available. These adapter molecules carry a single amino acid and are composed of RNA that serves as a physical link between the mRNA and the amino acid sequence of proteins. The ribosome then reads and identifies the mRNA strand's code, one codon at a time, and the complementary anticodon located on one end of a tRNA molecule is called attached. These codons provide the essential information needed to determine the specific related amino acid required. This process of a tRNA's anticodon attaching to the complementary codon on the mRNA strand occurs again as the ribosome continues reading the base sequence and the amino acids linked together covalently by a peptide bond. The first tRNA and its anticodon detach from the amino acid, leaving it behind to allow the continuation of the amino acid chain. The mRNA strand then moves through the ribosome as this procedure is repeated and its base sequence is read one codon at a time, and a complementary tRNA is called and bound, leaving behind its amino acid, therefore lengthening the chain. The amino acid chain is continuously built until the ribosome reads a stop codon on the mRNA strand and the amino acid chain is detached from the ribosome. A polypeptide has now been formed through this process, and the used mRNA is then expelled from the ribosome to dissipate in the cytoplasm. There can sometimes be issues in polypeptide synthesis, including misfolded proteins and their relation to neurodegenerative diseases. Protein misfolding is a common cellular event that can occur throughout the lifetime of a cell, caused by different factors, including genetic mutations, translational errors, abnormal protein modifications, thermal or oxidative stress, and incomplete complex formations. These misfolded proteins are believed to be the primary cause of a variety of neurodegenerative diseases such as Alzheimer's disease and Parkinson's disease.